welcome guys to class today. You might be at the pool or in your room or whatever, but this topic's going to be real interesting, and we're going to cover basically how does muscle contract, which is the fundamentals of this course. You can see me here. This is me working in my old lab, and basically we did a lot of stuff with muscle. You know, I did a biopsy study. You can kind of see how that looks. Basically, you actually stick a needle into someone's muscle and plunk out a chunk of muscle, which you see the right. We also did a lot of rat studies where we looked at muscle, and right there I'm grinding the tissue, extracting the RNA, and basically looking at gene expression in response to various training and supplementation protocols. So if you look at muscle, one of the things that makes it unique is its cell membrane. And its cell membrane is called a sarcolemma. Basically, what's cool about a sarcolemma is that it's excitable. What that means is that it can respond to various stimuli. Now, if you look within a muscle cell, you can see that it has several of these organelles, which are called myofibrils. You see the myofibril down to the lower mid-right. Okay, and what a myofibril is a long cylindrical organelle that has the capacity to contract. So each myofibril is comprised of repeating units called sarcomeres. We call sarcomeres are the basic unit of skeletal muscle contraction. So we look here. How is it? We see part of a myofibril here with 3 repeating units. Again, those are sarcomeres. Those 3 repeated units are held together by what are called Z-lines. And one Z-line connected to the next Z-line forms a sarcomere. Now, if we look deeper into a sarcomere, we see that a sarcomere is made of 2 filaments. You see the actin filament, which is thinner, and then the myosin filament, which is thicker. And you see the right, that's how we actually see it sort of on a microscope. You see how those zigzaggy lines, the Z lines, we see the myosin, which is those thick filaments in the middle, and then the actin. Those are our contractile proteins. Let's zoom in a little bit closer here. If you look at myosin here, you see why it's called the thick filament. You see it has a tail with a head connected to the tail. So those are the 2 units of the myosin molecule. Just think that's very, very important, okay? Because essentially that you have that tail and you have that head. That head essentially is flexible and can go backwards and forward. And it ends up binding to the actin molecule. Here's the actin molecule here. You basically see that you have these small globular molecules. And if you notice, each of those molecules has a little black dot. That black dot, that's binding sites. So what do you think binds to those binding sites? Well, if you go back here, it's the head of the myosin. Okay? So basically the head of the myosin binds to those binding sites. So you have G-actin, which is bound in a string forming F-actin, and then that sort of forms sort of a double helix. So the main thing you want to know is you have actin, and actin has several binding sites on it, and the head of myosin binds to those binding sites. So let's talk about the sliding filament theory of muscle contraction. The sliding filament theory basically states that the actin filaments at each end of the sarcomere slide inward on the myosin filaments, pulling the Z-lines toward the center of the sarcomere, and thus shortening the muscle fiber. Let's take a look here. It's a very more of a diagrammatical, animated picture here. So we see myosin. Basically, imagine the heads are connecting to the actin and sliding the actin molecules closer in, and then you see the contracted state down below. So again, the theory states that the actin filaments at each end of the sarcomere slide inwards on myosin filaments, pulling the Z-lines toward the center of the sarcomere and thus shortening the muscle fiber. Okay, so here we see that again. You see the thick filaments pulling the thin filaments inward. What do you also notice? That the Z lines come closer together. So you ever see someone who's super lean, right? And when they move their muscles, you kind of can see the striations in the muscles. What those stripes or striations are the Z lines moving closer to each other. So why are we not always contracting? Well, at rest, notice that the binding sites of actin are covered by something called tropomyosin. That's sort of that long, stringy molecule there. It covers up the binding sites. But you also have, on top of the tropomyosin, troponin. Troponin is sort of the molecular switch for contraction. So basically, at rest, the binding sites of actin for myosin to bind to are covered by tropomyosin. However, if calcium is released into the cell, it binds to troponin, which sort of acts as the molecular switch to contraction, because that pulls the tropomyosin off of the actin binding sites, allowing myosin to bind. We can see that here in cross-section. See the myosin head can't bind. Calcium is released into the cell. That binds to troponin, causes a conformational change, which pulls tropomyosin off of the actin binding sites. And now, basically, the myosin head can bind. So where do we actually get the calcium from in the cell? Well, it turns out we have calcium sort of storage bins in the cell. If you look down there at the myofibers or myofibrils, they're covered in that sort of whitish blue network. And that network is called the Sarcoplasmic Reticulum. Now, the Sarcoplasmic Reticulum is sort of a storage bin or storage bins for calcium. If you can get the calcium to be released from the storage bins, they'll bind to troponin, pulling tropomyosin off the actin binding sites and allowing myosin to bind. So how does this all get set up? Well, at rest, muscles are negatively charged. And changing their resting charge to a positive charge is what triggers the sarcoplasmic reticulum to release calcium. So how does that actually occur? Basically, if you look at a neuron, which is called a motor neuron releases what's called acetylcholine. When acetylcholine binds to a special place on the muscle called the motor end plate. When acetylcholine binds to the motor end plate, basically that causes sodium, which is a positive charge, to fly into the cell. This rapidly triggers a change in the charge of the muscle from negative to positive. So basically you have these sodium channels that open up and allow sodium to fly into the cell. When the motor neuron releases acetylcholine, and that binds to the motor end plate. So basically acetylcholine triggers sodium channels to open up. Now sodium can go into the cell. So once that happens, once we have this change of potential here, basically you have what's called an action potential. And an action potential is when you change the charge from negative to positive, and that charge gets carried first along the cell membrane, and then it goes into the cell, basically, and triggers the sarcoplasmic reticulum to release calcium. Now, to move from the cell membrane into the cell, we have these structures called T-tubules, which you can see there, right? See the two tubules basically carry the charge into the cell, releasing calcium. So again, calcium is the key to muscle contraction. It exposes binding sites to actin. So we have actin covered up. Calcium is released into the cell. That binds to troponin, pulls tropomyosin off the actin binding sites, and now contraction can occur. So let's sum this up here. ACH binds to the motor end plate, causing depolarization, or that rapid change of charge from negative to positive. The action potential is propagated to travel across the sarcolemma and then inside the muscle cell via the T-tubules. The change in membrane potential leads to the opening of calcium channels in the sarcoplasmic reticulum, causing calcium to rush into the sarcoplasmic reticulum. This binds the troponin, allowing tropomyosin to be pulled off the actin binding sites. And now, what happens? Now, myosin can bind to actin, and it slides it over itself. Here's a look at what that happens. You see at the top, you have the attached state of myosin to actin. What myosin does is it has an enzyme called myosin ATPase. Now that breaks the myosin, it breaks ATP, which cocks the head back. Then it's in a higher energy state, and it basically flings forward, binds to the actin, and slides it forward. So basically, again, what's going on is that you split ATP, that cocks the head back, it then comes forward with a lot of energy, binds, and slides the head in. Almost like basically when you're rowing through water. So again, I said what you need to know. After actin sites are exposed, myosin can bind to actin. Myosin ATPase, and the myosin head splits ATP, which puts the head back in a cocked state. The myosin head flexes and performs a power stroke, which slides the thin filament over itself. This moves the lines closer. To break the attachment and start over again, ATP must again be broken down to ADP. Last topic today is muscle fiber types. So before I go here, let me ask you, what are the different muscle fiber types we have? Okay, some of you said fast twitch, some of you said slow twitch, some of you said type 1, some of you said type 2. You know, you're all essentially right. So we look here. We have type 1, which is a so-called slow twitch. We have type 2A, which is sort of a hybrid and intermediate. It has fast twitch properties, but it's more conditioned, so we use a lot of oxidative properties. And then we have type 2X, which is fast twitch glycolytic, which means it's not very endowed with oxidative capacity, but it's very large. So if you look at maximal shortening velocity, you can see that type 1, 2A, and actually it's 2X, should be 2X there. The velocity or speed of contraction is faster as you go up the wire. Now here's a table. You can memorize this table, that's fine, but I like to do this intuitively, okay? Say you were a designer and you were designing type 2A, type 2X, and type 1 fibers, all right? Your slow twitch fibers, or type 1, are meant for aerobic capacity. So if you were to design something for aerobic capacity, what would you do? How big would the fiber be? It'd be small, right? You don't need to provide a lot of extra oxygen for that tissue, which is good for endurance. How rapidly would it contract? It would be slow, right? So that's why we call it slow twitch. You know, how much myoglobin would it have? A lot, right? How would it get its primary mode of ATP? Aerobically, right? So these are the things that you want to think about. How much mitochondria and capillaries would it have? A lot. Now, if you want an intermediate fiber, that's the type 2A, everything's going to be intermediate. If you have a fast, big, brute fiber, it's going to be extremely large. It's going to be primarily glycolytic and ATP-PC. It's going to go for a short duration. It'll be very fatigable. Okay, so that's how you kind of remember these, is just think of it on that perspective, right? So on the exam, you know, think about this. Think about type 1 and type 2 fibers in general. If you are a power lifter or bodybuilder, what type would you want to be born with? Okay, for you guys, for you guys who said type 2, you'd be correct. If you want to be an elite endurance athlete, what would you select? Yeah, the slow oxidative fibers, right? So that's very important. Now the real question becomes this: how malleable is this? How can you manipulate this with training? Right? Are we genetically programmed to have a certain dominant fiber type, and can we change that? Well, that is a real interesting topic. What I can tell you is that we'll talk more about that in adaptations to skeletal muscle. And what I can tell you is that it seems that early on in training, definitely when someone is young, that it is malleable. You can manipulate it more than when you're older. However, we will talk about possible training variables on how to manipulate that, and that it may be possible to occur. All right, thank you for your time, guys. And basically, there's going to be your quiz will be online on Sunday, and we'll talk about this in class as well. Also, make sure you see the written up questions for this. Talk to you later, guys. See you in class. Bye.